Good evening, good afternoon, good morning, wherever you are on the planet. Welcome. I'm Dan Bernard from McGill University and happy to welcome you to the third lecture in SSR's Neuroendocrinology of Reproduction webinar series. Before I introduce our speaker, I want to thank Pete Hansen and the SSR for organizing this series. I think it's indicative and emblematic of SSR's commitment to this part of the field. And I really want to encourage everyone to consider joining the society if you're not currently a member. I am a neuroendocrinologist. I am a longtime SSR member. If you have questions about what it's like to be a neuroendocrinologist in this society, I'm happy to answer any questions you might have. With that, it's my pleasure to introduce our speaker today, Dr. Victor Navarro, from the Division of Endocrinology, Diabetes and Hypertension at the Brigham and Women's Hospital, Harvard Medical School in Boston. I think the key for a moderator is not to talk longer than the speaker, so I just want to hit a few career highlights for Victor, just so everyone can learn a little bit more about him. He earned his PhD in physiology in 2006 at the University of Cordoba in Spain, where he worked with Dr. Manuel Tena-Sempere. He then did postdoctoral work from 2007 to 2011 at the University of Washington in Seattle with Bob Steiner. He returned to Spain for about a year or so before moving to Boston, where he's been ever since. First as an instructor, then an assistant professor, and as of 2020, an associate professor of medicine in Harvard, or in the Harvard Medical School. Victor's a longtime member of the Endocrine Society, the Society of Neuroscience, and he's now a member of the Executive Council of the newly formed Pan American Neuroendocrine Society, and we hope he's a future member of SSR. He's a great citizen within the field. He does a lot of service. He's a paper reviewer, including for BOR, grant reviewer. He's now a standing member of the BNRS study section at NIH and an associate editor for Neuroendocrinology. Victor's won many awards and has been recognized for his research, including a Fulbright scholarship and a Marie Curie fellowship. His program is very well funded currently by NIH. He's very productive with more than 80 papers and reviews in outstanding journals, including Endocrinology, JCEM, JCI, Journal of Neuroscience, and eLife. His lab is interested in the neuroendocrine control of reproduction and metabolism. And as you can see from his title here, he's interested in how these systems talk to each other, how metabolic cues can regulate reproductive physiology and behavior. So with that, I'll turn it over to Victor, who will tell us about novel bidirectional pathways in the hypothalamic control of reproduction and metabolism. All yours, Victor.
All right. Well, thank you very much, Dan, for the very nice introduction, and thank you very much for the invitation to speak this seminar series. So, like you all heard, I'm going to talk about the interaction between reproduction and metabolism in the context of the hypothalamus. So, I think we all agree that having a baby is a milestone, is a lot of joy and a great moment in life for anybody. But I'm sure also that if I ask any new parent in the audience what is reproduction, they're going to say that reproduction is exhausting. And it is true that there is a lot of energy that goes into reproduction, from finding a partner to pregnancy to raising a child later. And if we think about what happens in the wild, this is even more true, because usually in the wild, offspring comes in sets of many. So it really makes sense that throughout evolution, vertebrates in general actually evolved to have some sort of fail-safe mechanism that prevents reproduction from happening when we don't have enough energy reserves. And a good example of this is what happens in cases of eating disorders or in athletes that undergo excessive exercise. Usually in these patients, we find what we call functional hypothalamic amenorrhea, in which the whole reproductive axis shuts down because of the lack of energetic reserves. So how does the metabolic state affect reproductive function? This is one of the main broad questions that we have in my lab. And before getting into all the details, I want to make sure we're all on the same page. And I'm sure we are, given the audience of SSR. But just briefly as a reminder, when we're talking about the hypothalamic-pituitary gonadal axis or the HPG axis, we can say that one of the main players is GnRH neurons, which project to the median eminence, and they release GnRH, which gets to the gonadotropes and makes them release gonadotropins that in turn go to the gonads and induce the release of sex steroids that go back and feed back onto the hypothalamus to complete the gonadal loops. But in addition to GnRH neurons, we have KIS1 neurons, so different sets of KIS1 neurons. And here I'm focusing on two main populations, one in the anteroventral preventricular nucleus, which is mostly specific of the female and controls the pre-ovulatory late surge, and the other one that is common to both males and females, located in the arcuate nucleus, that is very particular in that it also co-expresses neurokinin B and dynorphin, and through the interaction of these neuropeptides, what we have is a pattern of pulsatile release of kisspeptin that then is mirrored by pulses of GnRH and LH. So these neurons, also called candy neurons, are considered right now to be the GnRH pulse generator. But of course, these neurons are not just by themselves, they're in the hypothalamus. They receive a lot of signals that determine the pattern of kisspeptin and GnRH release. Among those are these metabolic signals that I have here: insulin, ghrelin, leptin. So for the purpose of this talk, I wanted to focus on the role of a leptin to control not only metabolic function, but reproductive function. So the main question is, how does leptin transfer the knowledge of the available fat storage from adipocytes to the brain that then leads to successful reproduction? And to start this story, I think we should begin with understanding or trying to identify which populations of neurons actually are playing this role. And we know that leptin receptor is expressed both in glutamatergic and GABAergic neurons. So a few years ago, some colleagues, this is Bratlaus lab, started to look at the metabolic role of leptin receptor in these two populations, which is interesting here. You can see on the left, GABAergic neurons, and on the right, glutamatergic neurons. They're almost like a negative image of each other. And when we look at the arcuate, which is the area that we really care about, where these neurons that I told you are located, this is a pretty mixed population. So in our lab and others, we continue this study from the metabolic action to look at the reproductive action, and removed leptin receptor from both GABA and glutamatergic neurons. And as you can see here, when you remove leptin receptor from GABAergic neurons, you have an increase in body weight, but also this increase happens when you remove leptin receptor from glutamatergic neurons. And this is true in females and in males. Several studies later tried to do the opposite thing, which was to reintroduce leptin receptor in GABAergic or glutamatergic neurons. And as you can see in these images, if the black line is the control group, the red line is the DB mouse, and here is what happens when you introduce the leptin receptor into GABA neurons. So there is an improvement in body weight, but these animals don't look like controls either. And so what we can take from this is that leptin action needs to also happen at the level of glutamate neurons for normal reproduction to happen and for normal control of body weight. There are a number of studies looking at what those other GABAergic neurons are, and we can include and talk about AGRP for a long time. But the main reason why I'm focusing here on the glutamate part of it, which, as I showed you, is important in the control of body weight, but also reproduction, is because KIS1 neurons in the arcuate are glutamatergic as well. This is a study from our collaborators, Martin Kelly and Olivia Ronicliffe, showing that the majority of KIS1 neurons in the arcuate are actually glutamatergic. So how are KIS1 neurons regulated by leptin? Since the very early days of kisspeptin, we know that Kis1 neurons actually express leptin receptor. This is coming from Steiner lab in Seattle, where I was a part of years ago. But also we know that leptin can activate Kis1 neurons. And this is again another study from Martin Kelly and Olin Verniclip. So there is evidence that Kis1 neurons express leptin receptor, and the leptin can activate these neurons, which, like I said, are glutamatergic. Sometime later, Jose Donato, when he was at Carol Ay's lab, they did a phenomenal study in which they tried to determine the role of leptin in Kis1 neurons at the timing of puberty onset. In essence, what they found was that males and females are still fertile when you remove leptin receptor from Kis1 neurons. And at least in females, the timing of puberty onset was not different. However, there was not a clear assessment of what happens in the males or metabolic challenges that could determine exactly if there is a more subtle effect of leptin receptor or leptin signaling within Kis1 neurons. So in this context, then we ask ourselves, can Kis1 neurons be a direct target for metabolic regulation, in this case for leptin or any other metabolic factor? So in the lab, Sylvia Leon and Raja Talbi started looking at possible factors within Kis1 neurons that may be playing this role. And we identified, using a data set of RNA-seq from arcuate neurons in male and female mice, that the transcription factor NHLH2 is highly enriched in Kis1 neurons. And this factor is a basic helix, loop helix factor, and is highly specific of not only the arcuate, but also the whole brain in adulthood. As you can see here, this is the location where Kis1 neurons are located, and this is the expression of NHLH2. So it's very specific of Kis1 neurons, and we validated this using in situ hybridization. This is RNA-scope, and as you can see here, this is what happens in the male arcuate. There is a high degree of colocalization. In the female arcuate, there's colocalization as well. This is a little bit less true in the ABPB in females. And we can see this summarized here. So males, the majority of Kis1 neurons in the arcuate in the male do express this transcription factor, not so much in females, and definitely less in the ABPB. So the next obvious question was, well, does NHLH2 actually activate Kis1 neurons and TAG2 promoters? And I have to say that TAG2 is the gene that encodes for neurokinin B that I mentioned earlier. Is a co-transmitter of with kisspeptin within Kis1 neurons in the arcuate. So the first thing that we did was to identify the predicted sequence that NHLH2 binds to, which we did using a Jasper matrix. Then, with the help of Alejandro Lomnitzy in Portland, we identified a number of potential binding sites within the human, mouse, and rat promoters for Kis1 and for TAG2, which, by the way, in humans is called TAG3. So if you hear me say TAG2, TAG3, we're actually referring to the same gene in rodents or in humans. So the next thing that we did with his help was to determine if NHLH2 binds and activates these promoters, and this is a luciferase assay, and you can see how there is dose response effect of the presence of NHLH2 in the activity of both Kis1 and TAG3. And when we did chromatic immunoprecipitation, we found that indeed this transcription factor is recruited to the promoters of these genes. So it is highly expressed in Kis1 neurons, and we can activate Kis1 and TAG3 promoters. So we wanted to understand what happens in a mouse in which we remove this transcription factor from. And we created, obviously, like I said, this mouse. We did a reproductive phenotyping of females and males. Here's females first. And when we characterize reproductive phenotyping of females, we look at a vaginal opening as a sign of puberty onset. As you can see here, there's really no difference in the timing of puberty onset or first estrus, which is another sign of puberty onset. However, there was a slight disruption in the regularity of the estrous cycle. When we looked at the pre-ovulatory LH search, there was no difference in the controls and the knockout mice. So females have a very subtle phenotype. However, when we looked at males, this was not the case. These males were more affected, and you can see here how there is a delay in the timing of puberty onset, which is determined by preputial separation in the male. And this was not because of changes in body weight. As you can see here, body weight was the same. We even challenged these mice with half-fat diet, but there was no difference. So this is interesting because that delay that we were seeing in puberty onset actually correlated with a decrease in the expression of KIS1 that we can see here. This is through RT-PCR, but also through RNA-scope. You can see how it is clearly less in the KIS1 and HLH2 knockouts compared to the controls. So the interesting thing about NHLH2 is that it's regulated by metabolic cues. You can see here what happens when you deprive a mouse from food. The expression decreases, and this goes back up when you give food back to the animal. And apparently, most of this action happens through leptin, because we can recapitulate this effect when we add leptin. This is work that David Good has been doing, working with this transcription factor for a long time. We also know that leptin decreases very fast, very quickly when you fast, when you put this a mouse under fasting protocol, and you can see here. So we figured that if this is true in this mouse model that we were working on, if we fasted the mouse, we would have a more severe phenotype in terms of gonadotropin release. And this is exactly what we found when we fast the animals for up to 24 hours. This is the knockout animals. The LH decreases even faster than in the controls. However, when we refeed the animals, they both go back up. However, in this case, like in the paper that I just showed you earlier, we cannot determine if this action is depending on leptin or not. So to address that question, we have some animals that were fasted and then treated with leptin, and we argued that if leptin is acting through NHLH2, and there's some evidence that we didn't play here, but there's some evidence that NHLH2 interacts with STAT3, then these animals will not respond as well as the controls. And this is exactly what we found. So to summarize this part, we can say that the transcription factor NHLH2 contributes to the activation of KIS1 neurons in the presence of sufficient energy reserves by transmitting the permissive effect of leptin. So that part was addressing how KIS1 neurons themselves can use leptin receptors, or probably other receptors, to directly detect and respond to metabolic factors. But the next question that we have was, is there an indirect regulation for upstream neurons? Like I said, these neurons are not just alone in the hypothalamus, receiving connections for a multitude of other parts of the brain. So the first thing that I wanted to mention, and I'll go through this quickly because this has been published, is what we've been studying and finding about the pituitary adenylate cyclase activating polypeptide, or PACAP. So it was known for some time that PACAP receptor and PACAP knockout mice have disruption in terms of fertility. They were subfertile, they produced fewer number of pups. So Rachel Ross in my lab wanted to address, all right, where does this happen? Is there a correlation between the expression of leptin receptor, looking at STAT3 and PACAP? So she looked at different parts of the hypothalamus, and the area where the expression was highest is, as you can see here, was the ventral premammillary nucleus. So we started looking at this, and we wanted to address the question of whether PACAP mediates the reproductive role of leptin because of that specific co-expression that I just mentioned. So the first thing that we did was to create a leptin receptor PACAP knockout mouse model, and we did a similar reproductive phenotyping that I just mentioned, looking at vaginal opening and first estrus. And as you can see here, there is a very clear delay on both the days or the time of vaginal opening and first estrus. However, when we characterized the males, this was not true. There was no difference in preputial separation. When we looked at estrous cycles, there was also a disruption. They were having fewer estrous cycles in a period of 30 days, with obviously having longer cycles, per cycle, more days per cycle. And when we did a fertility test, these animals were having fewer pups. Moreover, when we also repeated a protocol to induce a pre-ovulatory GnRH surge, we observed that these animals have a disrupted ability to induce a pre-ovulatory GnRH surge, which could explain the fewer pups that we were seeing. And when we remove the gonads, we usually get a compensatory rise of LH, and there was indeed a compensatory rise, but this was significantly lower than what we find in controls. So if you remember what I showed you, the main area where this colocalization was happening was the PMV. And I want to again show this paper that I mentioned earlier, talking about the PMV is something that Carol Elias has focused for some time, and has a really great work documenting the role of PMV in the role of leptin. So we said, well, if this is true, then if we remove PACAP only from the PMV using the PACAP flux and injecting an AAV3, we should recapitulate the phenotype that we just saw. And this is exactly what we did. This is just here confirmation that we removed PACAP from this area. And as in the previous case, we saw a disruption in the estrous cycle. And as you can see here, summarized in these figures, and these animals also take longer to get pregnant. So the following question that we had was, well, do PMV PACAP neurons also project to the arcuate or the AVPV? So to do this, we injected in the PMV of PACAP cremized these viruses that can label projections. And as you can see here, this is in green KIS1 neurons in the AVPV or the arcuate, and in red projections that come from the PMV from PACAP neurons. And the answer to that question was yes, these neurons can project to both the arcuate and the AVPV populations. So you can have projections, but that doesn't mean these are going to be synaptic connections. So this is obviously the next question that we had. And this is work that we did in collaboration with Brown Lowell, using Chanelrhodopsin and then optogenetics to do what is called CRACM. And when we looked at the activity of KIS1 neurons after activating PACAP terminals in the arcuate, we saw that the majority of them were actually activated, they were responding. Some of the neurons in the arcuate that were not KIS1 neurons were responding as well, telling us that maybe PACAP has other additional roles in the arcuate that we don't know about yet. But when we looked at the AVPV, this activation was even more specific. The majority of the KIS1 neurons responded, and very few of those that were not KIS1 neurons responded. So the conclusion from that part could be that PACAP neurons in the PMV regulate reproductive function by directly modulating the activity of KIS1 neurons in the arcuate and the AVPV. So that was one example of indirect regulation through metabolic cues down to KIS1 neurons, and therefore the whole HPT axis. Another important part when we talk about leptin is melanocortin signaling. There's a number of studies that have documented how leptin can affect the activity of both AGRP neurons and POMC neurons. And when it comes to these two types of neurons, just a brief introduction for those that are maybe not very familiar with them. AGRP neurons are called hunger neurons, and they have two different kinds of action. One that is long-acting and goes through AGRP and neuropeptide Y, inducing, like I said, this hunger feeling, induces food intake. And another one that is short-acting, more fast, that goes through GABA release. But we also have two other different kinds of populations in the arcuate that induce satiety. One of them is the pro-melanocortin neurons, or POMC neurons, which have a long-acting that goes through alpha-MSH. And then there's another population that's not very clear which population it is, that act in a short-acting through glutamate to regulate food intake and induce satiety. So in this context, we wanted to know what happens with KIS1 neurons. We know that these neurons are intermingled with the other ones, and Are they a target for melanocortins or are they not? So in this context, what we know is that when we don't have MC4 receptor, which is the main receptor for alpha MSH and for AGRP, what we have is a continuous feeling of being hungry. And this is just an example here. These are two siblings. On the left, you're seeing a patient that has a mutation in MC4, and here's the sibling with no mutations. The body weight differences are very clear. And this is the same thing in mouse models. MC4 global knockouts have a significant increase in body weight compared to control mice. And when it comes to reproductive phenotyping, even though for humans, there hasn't been a complete definitive description of whether there is an actual reproductive phenotype or not, in rodents, this is very clear. And it usually shows up with changes in estrous cycle, ovarian morphology, and secretion patterns. And these are just examples of changes in the estrous cycles in MC4 knockout mice. And what happens in the ovaries, you have signals here of or signs of corpora lutea, which are signs again of ovulation, and nothing really or very few in the global knockout. So remember this because I'm going to refer to this in a few slides. So in the lab, Roger Talby, one of the postdocs, has been working to characterize what happens with Kiswan neurons and melanocortin. So the first step obviously was to understand and to assess whether Kiswan neurons express the melanocortin receptor 4. So through in situ hybridization, she found that indeed the majority of Kiswan neurons in the arcuate and in the AVPV do express MC4, and you can see it very clearly here in these images. So now that we know that Kiswan neurons can potentially be a target for alpha MSH and AGRP, the next question that we had is, like in the PACAP story, are these POMC neurons projecting to Kiswan neurons? And this is actually something that had been already shown in the literature. So there was clear evidence that indeed POMC neurons can project to Kiswan neurons, but there was not much more done. So we followed this up, asking also if these are synaptic connections. And in collaboration, again, with Martin Kelly and Olin Cleave in Portland, the first thing that we did was to assess whether Kiswan neurons in the arcuate can respond to alpha MSH. And as you can see here in this image, the answer is yes. Alpha MSH induces a very clear inward current in these neurons in the presence of TTX, so it's a direct action. So with their help, we also asked whether activating terminals of POMC neurons can lead to activation of Kiswan neurons. And the answer was also yes. Actually in the presence of a low frequency activation, optogenetic activation, we can see an effect that is glutamatergic. And when we have a high frequency activation, we see slow currents that are a response in the presence of neuropeptides. So obviously we wanted to know whether this neuropeptidergic activation was mediated by alpha MSH, and what we did was to repeat it in the presence of an alpha MSH, or sorry, an MC4 antagonist, which is called SHU 9119. And this activation, as you can see here, is very significantly blunted. So this was addressing the question that yes, POMC neurons can activate durally Kiswan neurons through the release of alpha MSH. So is the reproductive phenotype of MC4 knockout mice due to their obese phenotype or to deficient POMC to Kiswan signaling? We know that obesity itself can be a problem when it comes to reproduction. It might have very significant impairments from like the control of Kiswan neurons down to the gonads. So the first thing that we did to address this question was to generate a Kiswan specific MC4 knockout model. We looked, like in the other models, at vaginal opening and for estrus, and what we found here is that these mice actually had an advancement in puberty onset when we looked both at vaginal opening, and first estrus. And this, in a way, makes sense. We initially were expecting a delay. When we got this, we went back to the literature, and what we need to remember and remind ourselves is that MC4 is receiving also input from AGRP neurons. So when we don't have AGRP neurons, this is exactly what Stephanie Padilla and Richard Palmer published a number of years ago, that AGRP neurons can inhibit KIS1 neurons. And this is what I think we are having here. And this is the reason why these animals have advanced puberty onset. When we looked at the estrous cycles, what happened here is that we found a complete disruption of the estrous cycles in the KIS1 MC4 knockout mice. And this is summarized here. As you can see, the animals spend more time in diestrus than in the other phases of the estrous cycle. And when we looked at DLH search, this was also blunted, as you can see here. And when we looked at the ovaries, they looked very much like the global knockout ovaries that I showed you earlier. Compared to the controls, there were very few, if any, corporal lutea, indicating that these animals don't ovulate that well. And when we look at the number of pups that they have, as you can see here, they take longer to get pregnant and they have fewer numbers of pups. So it's interesting that these mice, despite being lean, because these females are not obese, they have the same reproductive abnormalities that the MC4 knockout mice do. So if that's true, we wonder, well, what happens if now we do the opposite thing? If we reintroduce MC4 in KIS1 neurons in an otherwise MC4 knockout mouse. And this is what we are doing in the lab. We took advantage of this model, which is a TB, a transcription blocker model, crossed with our KIS1 pre-mouse model to activate MC4 only in KIS1 neurons. And this is what you would expect, and this is what we got in an example of a control mouse for the estrous cycle, and an example of a typical MC4 knockout estrous cycle. And this is what happens when you reintroduce MC4 in KIS1 neurons only. The estrous cycle is rescued. And the interesting thing about this is that we are able to rescue the reproductive axis, even though we're finishing up the characterization of ovarian histology and other factors. This improvement of the reproductive function happens despite the fact that these animals are obese. So in terms of the metabolic phenotype, these animals look very much like the MC4 knockout mice. However, we are improving the reproductive activity. So this is basically what I just mentioned here. So the conclusion of this part would be the candy neurons mediate the metabolic regulation of reproduction of melanocortins through their direct action on MC4, and it's not because of their obese phenotype. So the next question that we had is, well, you saw all this, and this is how the reproductive axis actually receives all this information about metabolic cues, the metabolic state of the organism. That can KIS1 neurons actually do the opposite? This is the part of like the bidirectional thing that I included in the title. We know that this is a case in different phases, mostly more evident in women after menopause. There's usually an increase in body weight, let alone at the time of pregnancy. So there are situations in which we know that the reproductive axis can drive changes in the metabolic state of the organism. And a few years ago, Sasha Kaufman's lab in San Diego published a paper showing that impaired expectin signaling actually leads to an increase in body weight. So there is already some evidence in that regard. We wanted to explore this a little bit more. So the first thing that Raja in the lab has been doing to address these questions was to ablate KIS1 neurons in the arcuate and see what happens with the metabolic phenotype of the mice. And as you can see here, the ablation of the neurons happened using an AAV that expresses caspase-3. obviously accrue dependently. You can see an example of how these neurons are ablated. And when she monitored the body weight of these animals, you can see how there's a very significant increase in body weight over the period of time that these animals were monitored. So the answer to that question was yes, and this actually was in line with the study that Stephanie Padilla had published not long ago in females after silencing the same group of neurons. So we showed that this is also the case in the male. So what happens with the when we activate these neurons in terms of food intake? This is an aspect that has been controversial for some time now, because we've been trying to we've been trying to characterize exactly how these neurons control food intake. And there's been a number of studies showing that sometimes kisspeptin can decrease food intake, some other times it doesn't do anything. So one of the ways to address this was to use, in this case, direct chemogenetic activation of the neurons. So the animals were injected and then they were put in metabolic cages, and the food intake monitor after CNO injection, which is what we have here. And as you can see there, in green is animals injected with dreads. When these animals have Kiss1 neurons activated, the food intake, even though it increases, the increase is significantly lower than what happens in the control mice. So again, suggesting that Kiss1 neurons, this does not say that it's kisspeptin, but there's something coming from Kiss1 neurons, is playing an anorexigenic role. So if this is true for food intake, what happens in terms of energy expenditure? And one way of looking at energy expenditure is looking at the activation of brown adipose tissue. There is a brown adipose tissue pad right in the interscapular area in the back of the mouse. And so we implanted these transponders in the back of the mouse, and we repeated pretty much the same protocol that I just showed earlier. And what Raja found was that when you do this, you have an increase in the temperature of the brown adipose tissue after CNO injection. Again, suggesting that when we activate Kiss1 neurons in the arcuate, we're also inducing an increase in the energy expenditure of these animals. So I think with these two experiments, or three experiments that I just showed, it's clear that Kiss1 neurons regulate energy balance. But how does that happen? So one question that we had is, well, what are these neurons projecting to that could explain this phenotype in terms of energy expenditure? So using a virus that through mCherry and synaptophysin can label projections. These same Kiss1 Cre mice were injected in the arcuate, and then projections were assessed throughout the brain. And we found that the dorsomedial hypothalamus is an area that receives a lot of these projections. And as you can see here, these are fibers coming from Kiss1 neurons in the arcuate. So the answer to that question is, well, Kiss1 neurons in the arcuate do send projections to the DMH in these Kiss1 Cre mice. But we wanted to assess whether these neurons in the DMH are actually activated when we activate Kiss1 neurons. So using, again, dreads, what Raja found was that when you expose these animals to CNO, the activation of these neurons here in the arcuate leads to CFOS expression in the dread-injected mice in the DMH, but not in the control injected with mCherry only, no dreads. So there is evidence that there is some synaptic connection and activity coming from Kiss1 neurons in the arcuate to the DMH. And in this context, again, our collaborators in Portland recently published a paper in which they show that these candy neurons can actually project to DMH, and they do so, they activate these neurons through glutamate, because in the presence of a cocktail that blocks glutamate action, this is no longer true. This is your direct action. And moreover, they were able to identify that these neurons were actually leptin receptor positive neurons in the DMH. So we said, well, if that's true, what happens if we do the same experiment that I just showed you, but now we do it in KIS1 knockout mice? So there's going to be no kisspeptin around, but these neurons are going to be still present, and we have evidence that there's still projections from KIS1 neurons going to the DMH. And what happens if we activate the neurons with the same protocol? And the answer was that we also found that these activation can lead to the activation of neurons in the DMH. So this was telling us that even though it's not excluding that kisspeptin may have an action at the level of the DMH, there is something else that could be glutamate, according to Martin's and Olinas and Todd's publication. Could be glutamate. So this definitely deserves further investigation, and that's what we are working on. So what we know is that those leptin receptor neurons in the DMH have been described for a while now to activate brown adipose tissue through the RPA. So we wonder, well, if this is the case, what we're addressing here, what we're identifying is a pathway that goes from KIS1 neurons to eventually activating the brown adipose tissue. If we co-inject M-cherry in the arcuate of KIS1 neurons and then a retrograde tracer in the RPA, we should be able to see some co-labeling, some neurons in the DMH that receive both kinds of projections. And this is exactly what we did, and we found that indeed there's a population within the DMH that is labeled when you inject these retrograde tracers in the RPA, but also receives these projections from the arcuate. So I believe we are, or we have identified a pathway that starts in KIS1 neurons in the arcuate through glutamate and or kisspeptin, leads to the activation of leptin receptor neurons in the DMH, and eventually to the activation of RPA neurons and brown adipose tissue. So to go back to the model that I mentioned earlier in females, I'm sure some of you may be wondering what happens with the metabolic phenotype of those KIS1 neurons that didn't have NC4 in KIS1 neurons. So this is what happens in males. If you remember, females did not have any metabolic phenotype or normal child. However, when we looked at males, we were very surprised to see that on the normal child, this is no high diet. The males that don't have NC4 have a significant increase in body weight. And when we exposed these animals to high diet, this difference in body weight was even exacerbated. So when we did an MRI for these mice, we observed that the difference in body weight comes mostly from an increase in fat mass. And when we put these animals in metabolic cages, we saw something interesting. When we looked at food intake, we observed that the actual food intake, and this is in red, the knockout mice, it's not that much, it's actually lower than what happens in the control mice. However, something that is different is the circadian behavior of food intake. You can see here what happens in a control mouse. As we all know, mice are nocturnal animals, so they eat more during the night and less during the day. However, this rhythm is lost when you remove MC4 from KIS1 neurons. And it's interesting because this matches what Stephanie Padilla published in the female not long ago. When she silenced KIS1 neurons in the female, she also saw that total food intake is the same, but this circadian feeding behavior is lost. So there might be a mechanism, we don't know yet what it is, that is involved in KIS1 neurons in the circadian rhythms. When we looked at energy expenditure, we observed that these animals actually have reduced energy expenditure. They have lower CO2 production, lower oxygen consumption. And we figure, well, if this is true, the animals have lower energy expenditure. One of the ways, it's not the only one, but one of the ways in which we can control energy expenditure is, like I mentioned earlier, through brown adipose tissue. We had evidence already that we can activate brown adipose tissue when we activate KIS1 neurons. So what happens in these animals. If this is true, we should see a difference in at least UCP1 uncoupling protein 1, which is one of the main markers in the brown adipose tissue of activation. And this is exactly what we found, that these mice have lower UCP1, suggesting that there is lower activation of the brown adipose tissue. So with this, what we think we have here is a mechanism in which POMC neurons release alpha MSH that can potentially bind MC4 on KIS1 neurons. And these KIS1 neurons are going to contact leptin receptor neurons in the DMH that in turn activate RPA neurons to induce the activation of the brown adipose tissue. So overall, I think the conclusion for this could be that KIS1 neurons are active players in the control of energy balance and mediate the action of melanocortins on energy expenditure. And the overall conclusion would be that KIS1 neurons receive metabolic information through indirect upstream neuronal networks, as well as durally through transcription factors sensitive to the energetic state that ultimately adapt GnRH release to the level of energetic reserves. And if you remember throughout the talk, I showed you that these mechanisms are actually sexually differentiated, and some are more predominant in males and some are more predominant in females. So with this, I obviously just want to thank first and foremost the members of my lab, who are the ones that have done all the work, and our collaborators and our funding sources. I'll be more than happy to take any questions.
Great, thanks, Victor. Excellent timing. So at this point, I'll just remind everyone that I will ask your questions for you. You need to put them either in the Q&A or in the chat. You can pick your poison about which vehicle you'd like to use to ask your questions. Maybe while we're waiting for the first question to come in, I can get us started. If I can find my cheat sheet here, because I was taking some notes. I guess in no particular order, and especially because you kind of came back to circadian disruption with that last model. I was wondering, when you were studying the what is it called, NH-LH2 regulation, it binds to E-boxes, which of course are also the binding sites for the clock genes. So I'm just wondering if your manipulation of those cells of NH-LH, if you think you might have screwed up circadian behavior within those cells.
That's a very good question. The short answer is we don't know, because we didn't find a metabolic phenotype in those mice. So we didn't put them in metabolic cages, which is the easiest way for us to determine whether there are changes in either locomotion or food intake. So we didn't look at that in detail, but at least in terms of metabolic phenotype, which is probably part of the reason why the KIS1 MC4 knockout mice are, at least the males, are more obese, that was not present in that model. So probably not.
Because I was just thinking about, you're basically removing the competition to some extent, with the knockout. But okay, interesting. So Jennifer Hill has our first question in the Q&A. Excellent talk. MC4Rs are involved with sexual behavior as well. Have you examined whether restoring MC4Rs on KIS neurons allows normal mating?
That's an excellent question. This is something that we have in our mind. We've been trying to do a first reproductive phenotyping with the most obvious markers that we can find, but we know that mostly in the amygdala, MC4 is very critical for sexual behavior, but we don't know if KIS1 neurons in the amygdala, which is another population that I haven't talked about, do actually have MC4 or not, but it could be also mediated through ABPV. I know Julie Baker has published a publication showing that KIS neurons in ABPV are involved in sexual behavior, and these neurons, 70 plus of them express MC4, so it's very likely, but we have not addressed that question yet. Good one, though.
Okay, thanks, Victor. The next question comes in from Richard Pierre. He says, great talk. Apologies if I missed it, but were the mice ovariectomized in your chemogenetic activation experiments? If not, do you think estradiol secretion after KIS1 neuron activation plays a role in the effects you saw?
Yes, excellent point. And we thought of that. And yes, the animals are gonadectomized and replaced precisely because of that reason. Because any manipulation of KIS1 neurons, we know that it's going to lead to changes in melanocortins and sex steroids, which can affect all these different parameters that we're looking at. So they were.
Okay, maybe I'll ask another one. I'm curious about the CRE drivers that you're using and how specific they are. So for example, in one of the stories where you were using the leptin CRE and you saw effects on LH, I couldn't help but wonder whether the manipulation may have been at least partly attributable to the knockout within gonadotropes. Because we know the leptin receptor is expressed there. I don't know if the CRE is also expressed there.
Good point. You're talking about the pack-up model, or
exactly? Yeah.
I mean, it could be. Some of the factors that we're looking at are very central. And for the pack-up model, for example, we tested the fitness of GnRH neurons, and it was fine. But when we activated KIS1 neurons, they were not. So I think we have evidence to show that there is a central phenotype clearly. Now, whether there is something else at the level of the gonads, we don't know. I cannot remember for sure if they do express pack-up or not.
Okay, it's just I think you're aware. We knocked out GS and the gonadotrope, and the post-gonadectomy increases in LH and FSH were blocked. And we don't know if that's because of an impairment in GnRH signaling or PACAP signaling as another candidate. So that's what made me think of it.
Yeah, that's a good.
So we have a question that's come in from Jurgitsa Kas. Great talk, Victor. For the KIS MC4R knockout and disrupted estrus cycles, do you think the surge is affected? Do AVPV KIS neurons also have MC4R, or is pulsatile LH I think maybe exclusively dysregulated? Have you assessed LH pulses? So I guess she's asking about the relative contributions of the two populations.
That's a great point. So the LH pulses that we have looked at are not unchanged, but the one thing that I didn't mention about this model with melanocortins is that there's a big difference in younger mice versus older mice. So one of the things that happens in the global knockouts is that they have very early reproductive senescence. So we're trying now to assess whether these KIS1 MC4 knockout mice at an older stage have an impairment in LH pulses. But like I said, in younger ages, they don't. However, we did look at LH surge, which I showed in one of the slides, and it's significantly blunted. So there's a little bit of LH release, enough to have some pups because they're not completely infertile, but it's significantly blunted because 80% of the KIS1 neurons in the AVPV do have MC4. So actually, we think that most of the reproductive phenotype that we're seeing is actually mediated by the action of alpha MSH on KIS1 neurons in the AVPV, while the metabolic phenotype that we're seeing in the male, that is not present in the female, is mostly due to changes in the arcuate population. Great question.
Okay, maybe I'll ask another one. So, and this is probably well known, and maybe I should know it, but when you did your tracing experiments, again, given that you're seeing a circadian, an effect on circadian behavior, at least in terms of feeding, like, did you see that those KIS1 neurons in the arc are projecting to the SCN? I mean, have you thought about how you're altering the timing of feeding?
That's a great point. We did not see any projection there, and I believe Stephanie Padilla has also looked at projections to the SCN, and there are not clear projections happening there. So it's either those neurons are receiving some circadian input from either the SCN or an indirect group of neurons, or is an intrinsic oscillator that is affected. Yeah.
Okay. Maybe if we go to the model where you were rescuing MC4R, I might have missed this, but what's the promoter that's driving the MC4R expression, and how does the level of expression relate to how the receptor would be expressed endogenously? And I guess the reason I'm asking that is, if I remember correctly, MC4R has constitutive activity. And so I wonder if you can discriminate between the effects of an overexpressed constitutively active receptor versus putting a receptor in that will now be responding to alpha MSH.
Good point. I believe it is CMV, the promoter, but once you remove the stop codon with the accretor combination, it's going to be constitutively active. So your point, it's a good one. We don't know. Yeah.
Okay. I mean, I assume it's constitutively active in all contexts, but I don't know if that's true or not.
I don't know if that's going to have a biological effect in the absence of alpha MSH. That's a good question, but we don't know.
Yeah. Okay. So a question has come in through the chat from Jacob Moeller. Sorry, Jacob, if I'm not pronouncing your last name correctly. Since you see MC4R knockout mice go into reproductive senescence earlier, and a degradation of circadian output signals are associated with the onset of reproductive senescence, do you think a decline in circadian signaling underlies the early onset of senescence observed in the knockout animals?
It could very well be. That's one of the hypotheses that we have, and something that we're trying to test right now with different cohorts of different ages to see if we can actually address this question. It's a good one. We've been asking the same thing ever since we found it.
Okay, we have someone who wishes to remain anonymous, but thanks you for your wonderful talk, and asks, could changes in metabolism lead to altered urinary metabolites and impact the hypothalamus through pheromones?
Good point. I don't know. That's a short question, a short answer. I don't know. It's a good point. It's probably likely, but I don't want to speculate because I don't know. Okay,
that's always a good answer when you don't know the answer. Richard has another question. There's a preamble. So we didn't see effects of melanocortin 2, I don't know what that is, but you probably know what it is, on arcuate kisspeptin neuron firing in contrast to the inward currents you showed in response to alpha MSH. Do you have thoughts on this discrepancy?
Yeah, I believe he's probably talking about the work that probably him with Ellen Harbison did. Something that is critical in the response of these neurons is the estradiol milieu. So I believe the paper that he's referring to, the animals were in diestrus, and these animals are estradiol replaced. And also there's a difference between cell attach and whole cell recording. So I believe those are the differences that explain the discrepancy. It's not like, it's just mostly estradiol milieu and the technique of recording and the two different experiments.
Okay, we're at 6, but maybe I'll ask one last one, going back to the first model, the NHLH2. You showed that there was a sex difference both in the expression and the effect of the knockout. So I guess sort of a low-hanging fruit question is, what explains the sex difference? And did you see changes in the expression of that transcription factor across the estrous cycle?
We haven't looked at the estrous cycle. We have seen changes throughout development. And to your question as to what determines that sex difference, we don't know. I guess one way of doing this would be to alter the sexual milieu early in development during the critical period and see if we masculinize or feminize the expression of this factor within Kiss1 neurons. We haven't done that, but I think that would be probably the best way of addressing that.
Okay, so you think it's actually something that's sort of an organizational effect during development rather than something that's activated in adulthood?
That would be my guess.
Okay. Okay, great. So we're at 6:01 and I don't see any additional questions that have come in through the Q&A or through the chat. So at this point, I'll thank Victor for a great talk and all the attendees for coming and for asking questions. And I'll just remind everyone that there's one last summit webinar in this series, which is a week from today. And I hope we'll see everyone there. So have a good evening. And Victor, thanks again.
Thank you so much.